So it's my pleasure to introduce Jesse Dixon, who's assistant professor at the Salk. Jesse did his MD PhD here at UCSD and did his PhD in Bing Ren's lab, and then was subsequently a faculty fellow at Salk, which is a quite prestigious position, and then has stayed on at Salk to run his own lab. The focus of Jesse's lab is 3D organization and chromatin and changes in organization of the genome in disease. And yeah, we're very happy to have you here, Jesse. So welcome.
Cool. Okay. So let me turn the mouse on. I guess I'll just use my mouse like this as a pointer. Okay, cool. Awesome. So thank you for the invitation and for giving me a chance to talk about some of the work that's been going on in my lab. And it's always kind of cool to come back across the street and visit. Yeah, so like Kyle said, my lab is interested in how our genomes are organized in 3D space. And really kind of a major motivation for this is actually trying to understand how genes are regulated by distal regulatory sequences in the genome. And so if you do kind of any variety of modern sort of genomic assays, you'll be able to identify putative regulatory elements fairly readily in the genome, which is kind of have in this kind of cartoon type diagram here. But inevitably, you'll come to a situation if you're doing this kind of studies where you identify a putative regulatory element, and from the one-dimensional sequence of the genome alone, it's not necessarily obvious sort of what is the target gene of a particular regulatory sequence. And so this has been kind of a major motivating factor for why we were interested in this in the first place, really to try to understand which enhancers and which regulatory sequences work on which genes. And I actually think sort of even beyond that, there's kind of interesting mechanistic aspects thinking about, you know, why an enhancer will work on one gene but not another, or what controls specificity, or if there's things like molecular compatibility between genes and regulatory sequences. And the reason we look at sort of how our genomes are organized in space is that sort of the prevailing models for how enhancers work is that they have to be brought into close physical proximity to the genes that they regulate. And this allows promoters that bind at the enhancer to interact, or proteins that bind at the enhancer to interact with proteins of the promoter, and that this can facilitate transcription. And so this idea of like looping as a model for how enhancers communicate to genes has actually been around for an extremely long time. So these are some reviews from Bob Tejan and Jim Katanaga here at UCSD, sort of explaining, you know, at the time, sort of the prevailing theories for how enhancers worked at a distance. And they all invoke essentially these kind of looping models to have physical communication between an enhancer and a gene. And this, I think, is also sort of based on structural studies of chromatin and DNA that literally date back for over, you know, since the 19th century, where people had observed sort of using different methods, microscopy, electron microscopy, that DNA or chromatin could exist in these kind of looped conformations. Is that?
How we minimize the Zoom?
Oh, okay. Okay. Cool. But yeah, so there's basically sort of structural evidence that you can form these kind of DNA loops in the genome. I think the thing that's actually kind of interesting looking at some of these older reviews is actually that one of the significant sort of outstanding questions is not necessarily were there loops in the genome, but how they might actually form. So what are the processes that will actually bring enhancers in close proximity to their genes? And that's actually something that I think we've learned a lot more about in recent years with the advent of sort of additional tools and assays to kind of study genome organization. And so we've been doing these kind of experiments for a while now, dating back to when I was a student in Bing's lab, where we use these Hi-C assays to describe features of our genome that we call topologically associating domains, or TADs. And the way that this really worked is, you know, the idea in these Hi-C experiments is if you're interested in finding which sort of genes and regulatory sequences are in close proximity, you can take a population of cells and you fix them with formaldehyde, and that preserves protein-protein and DNA-protein crosslinks. You can then digest chromatin and fill them in with nucleotides, one of which is linked to a biotin, and then ligate these ends together, which preferentially is occurring between sites that are in close spatial proximity. You can then isolate these ligated fragments and subject them to high-throughput sequencing to get a picture of sort of all the different sequences and elements that were in close spatial proximity. So we've been
sorry to interrupt. We're still seeing presenter view. we've said in chat several times, and it
Oh.
hasn't gotten noticed. So it's still presenter view on the slides.
So the way to do that
Is
is
that okay?
That's fine.
You
by
need
me.
the-
There we go.
Is that better?
No, I don't have notes, and
Okay.
I'll have to maybe have a few surprises in terms of what slides come up next. But
This
I
is
think-
good.
Okay. Are you
Okay,
sure?
yeah, okay. Cool. All right. So yeah, we've been doing these experiments for a number of years now, and this is what some of the high C data that we generated in these early years look like, where this kind of red signal is showing how frequently you're identifying different regions of the genome in close proximity. So this kind of triangle type region, I'm losing my mouse, this kind of triangle type region would represent a region of the genome that's about a megabase in size, that's forming lots of contacts with itself. This region would be a segment that's about half a megabase in size. It also forms a lot of contacts with itself, but the two are relatively spatially isolated from each other. And so we call this kind of patterning these topologically associated domains, or TADs. And sort of with more higher and higher resolution versions of these assays, it's also apparent that these kind of domains have these sort of focal peak-like signals at the corners, which is really indicative that they're likely forming through the presence of loops in the genome. So we also have learned a lot about what are the factors that give rise to these kind of TADs and loops in the genome, and they appear to really exist through an interplay between the cohesin complex and a DNA binding protein called CTCF. So cohesin is consists sort of this multi-subunit protein complex. It has sort of a core ring that consists of SMC1, SMC3, and RAD21. That also has an associated STAG, one of two STAG subunits associated with it. There's a pair of proteins, NIPBL and MAO2, that are considered sort of loader proteins. At least historically, they're thought that they load cohesin onto chromatin, though there's recent evidence that they also are critical for its process of actually traversing or extruding along chromatin. And cohesin is opposed by sort of two proteins. One is WAPL, which kicks it off of chromatin, and the other is CTCF, which doesn't kick it off, but it essentially stalls it from progressing along a chromosome. And the prevailing model for really how this works is through a process that's called loop extrusion. And so this is basically a cartoon of these kind of TADs and loops. And the idea is that cohesin will essentially bind to a piece of DNA and begin to form a small loop. And this will sort of progressively enlarge until it encounters a CTCF binding site, where CTCF is actually oriented in a particular manner such that the N terminus of the CTCF protein is sort of facing an oncoming cohesin molecule, and that somehow arrests this. This process has been observed in vitro using essentially purified subunits. It hasn't really been directly observed with microscopic methods in vivo, but there's sort of a significant amount of evidence that would support that this is one of the major mechanisms for how these kind of domains form. The other aspect that clearly also supports that these have a role is using high C studies and protein degron methods, where factors like CTCF or cohesin can be rapidly depleted. And you can see that these kind of domain and loop type formations are rapidly lost upon loss of these protein complexes, suggesting that these are really playing major roles in how our genomes are organized. And so while we've learned a lot about what the factors are that contribute to how kind of structures like loops form, it's actually been very confusing then trying to relate this back to these ideas of chromatin looping as a model for enhancer gene regulation, because the effects are actually quite contradictory or confusing. So in this paper from Erez, Liebman, and Aiden's lab, they used RAD21 to rapidly deplete, or they use auxin to rapidly deplete RAD21. You essentially eliminate all loops and TADs in the genome. But if you look at what genes change, there's only a tiny fraction of genes that actually change in response to this sort of acute cohesin depletion. And this is sort of in contrast to different contexts, where this is a paper from Matthias Merkenschlager's lab, where they have RAD21 knockout macrophages that they stimulate with lipopolysaccharide. And in this case, the sort of stimulated response is largely attenuated in the absence of cohesin. And so the idea here is that perhaps cohesin plays more of an important role in sort of transitions in gene expression as opposed to maintaining sort of steady state patterns. There's also been somewhat conflicting evidence when we look at things that are perturbing chromatin organization at sort of a genetic level. So there's this really beautiful paper from almost 10 years ago now from Stefan Lindlos's lab, where they had identified families that had these disorders of limb development. And interestingly, they all had structural variants that map to a similar location in the genome, but they don't really overlap with each other. But what they all do is they actually all disrupt the boundaries between TADs. And what this does is this causes enhancers that normally regulate this FA4 gene to begin to regulate genes at sort of different sites nearby. So this Wnt6, Indian hedgehog, and PAX3. So this is really cool. And this is actually a really, I think, seminal paper when this came out, because it really suggested that, yeah, these TADs and TAD boundaries can have important impacts on regulating phenotypes and on gene regulation. But there's also several studies that have looked, I think, at more broad sort of comprehensive sets of structural variants and actually found that structural variants have a really small effect for the most part on gene regulation and gene expression. So this bottom one is a paper looking at these balancer chromosomes, which are these highly rearranged chromosomes in Drosophila. And the top is actually a paper that we were involved in with Andy Futrial's lab, where we're looking at structural variants from 2,000 different patient samples. And when we do this, we can see that really only a small minority of structural variants actually lead to any kind of change in expression. And so, again, there's sort of this kind of conflicting nature where some instances seem to have large, significant impacts on gene regulation, and others have sort of minimal impacts. Okay. Okay, so in my mind, this kind of sets up this sort of conundrum, which is that, you know, these using sort of several different methods to sort of alter genome organization and 3D genome structure, we can see that there are some circumstances where there's really big impacts on gene regulation, but in many other cases, there's very, very little impact at all. And this has been kind of a major focus of our lab for quite a while now. And so I'm going to kind of tell you two stories that we've been working on to try to better understand this. So one is actually looking at gene reactivation during mitotic exit. And the second is looking at using CRISPR-Cas9 genome engineering to essentially create structural variants de novo and examine their impact. So I'm going to start with this mitotic exit part. Like I mentioned a second ago, you know, there's this paper from Matthias Merkenschlager's lab, and there's sort of other work that's been done in kind of related settings, where in the absence of RAD21, you sort of fail to induce these sort of new gene expression patterns. And so the idea is that, okay, well, potentially cohesin is more important for the sort of transitions that might occur in gene expression. But I think this raises kind of a lot of questions, or like, you know, why is it that these kind of inducible genes seem to be more sensitive to this? And what kind of transitions might actually be fit this kind of pattern? Like, is this the case for all different signal-inducive transcription events or whatnot? And so the strategy that we've actually used is to try to look at the activation of gene expression that occurs during the exit from mitosis. And so during mitosis, transcription is essentially entirely shut off. But in the kind of minutes to hours following exit from mitosis, you essentially reactivate the expression of essentially all of the genes in the genome that are expressed at least in G1. And of course, this has to be done sort of faithfully. You need to reactivate sort of the correct genes that are critical for regulating cell identity. And at the same time, there's actually a dramatic reorganization of chromatin that occurs during the cell cycle as well. Chromosomes are, of course, dramatically condensed during mitosis and then have to refold along sort of similar timescales during the exit from mitosis. And this is kind of an area that's also been fairly well studied over the years, thinking about this idea of what's called mitotic bookmarking. So the idea is, are there mechanisms that the cell would essentially inherit through the cell cycle to allow sort of faithful reactivation of the gene expression programs during mitotic exit? And so this is something that we thought would be really interesting, because this is really probably the largest transition in gene expression that a cell really ever undergoes. And to try to use this as a system to try to understand what genes might be sensitive to sort of cohesin depletion. And so the idea, of course, is cohesin contributes to the formation of 3D genome organization. And so we were hoping to basically use methods to perturb cohesin and then essentially watch chromosome structure reform and gene expression become reestablished either in the presence or absence of this. But it actually sort of wears two hats, so that the other thing that cohesin does is it also facilitates sister chromatid cohesion. So that's where its name comes from. These are sort of largely independent functions. But the challenge is that cohesin is actually required for cells to passage through mitosis. And you can actually see this in kind of dramatic examples. This is something that we did, it's kind of an entirely different focus, but I think it actually shows really nicely how cohesin is really required for the passage through mitosis. And so this is basically just imaging cells over the course of 24 hours. The cells have a degron on RAD21, but we're just treating them with a control DMSO in this case. And so you can watch this and you'll see the cells sort of divide. You see a mitosis, they sort of rapidly progress and exit through mitosis. This is happening over and over and over again. And then in the conditions where we add this DTAG molecule to degrade RAD21, so we add it at the same time that the movie starts. And so there's like one mitosis like right here that you'll see successfully completes this, but it is degraded over the course of about an hour, and you'll basically kind of see what happens. So that cell divides, but then you'll start to see these, like there's a mitosis here, and it's basically struggling and struggling, and it can't do it. It can't exit mitosis, and now it finally does, but it doesn't actually separate. So it basically undergoes this sort of mitotic collapse. So it basically quits mitosis effectively. The cells are actually 4N after this for the ones that actually progress through S phase. It's kind of amazing. But they really dramatically struggle and fail to really progress through mitosis. And so we wanted to avoid a situation like this. And so we needed to basically have alternative strategies other than degrading these kind of core components. The other thing that I would say is, this is just an aside, but I thought these were awesome videos, and we have 3 kids, and I wanted to teach them about like cells and cell division. And so I showed this to them. And then I showed them the wild type one first, and then I showed them the RAD21 degron where the cells are like really struggling to divide. And I told them like, oh yeah, Tessa in the lab made a mutation so the cells might not be able to divide. And she's like, why did she do that to herself? So yeah. Okay. So the strategy that we took is actually to use, is this on the screen? Okay. The strategy that we took is actually to try to target the protein NIPBL, which is considered to be the cohesin loading protein. And so NIPBL is responsible, or it's thought to be responsible for helping to put cohesin onto chromatin, but it's actually essentially switched out or replaced once sister chromatid cohesion is established. And it's thought to be essentially fully evicted from chromatin during mitosis, and so the cells can actually progress through mitosis without NIPBL, or they normally progress through mitosis without NIPBL. And so we put these FKBP12 degron tags onto NIPBL, and what this allows us to do is then, this is in just cycling cells, we can add these drugs, these small molecules, in this case, this DTAG V1, and this will rapidly and fairly completely degrade NIPBL like very, very quickly. So within an hour, we see most, if not all, of NIPBL is gone. We can also do this, again, this is just in cycling cells, degrading this and doing things like high C, and we can see that the loss of NIPBL here largely eliminates most kind of TADS and loop type structures that we see. And so using this, we can then try to use the system to essentially look at the reformation of 3D genome organization during mitotic exit. So what we do is we synchronize cells with thymidine in S phase, we release them, and then we resynchronize them in mitosis or prometaphase with nicotazole. At the same time, we treat them with either DMSO as a control or this DTAG molecule to degrade NIPBL for 2 hours. And then we release them from nicotazole, but keep them in the presence of either DMSO or drug, so they maintain the loss of NIPBL during the process of exit. And so we do this, we can do things like PI staining, and we can see that either the DMSO or the DTAG conditions essentially exit mitosis with nearly identical kinetics. So it doesn't look like these actually have this kind of mitotic delay phenotype that I showed a second ago for the RAD21 cells. But then this allows us to do experiments and to kind of see like what's going on during this process. So this is looking at wild type conditions. This is essentially in the synchronized cells that are in prometaphase, and this is over 2 hours of release, and you see the reformation of these domains. And we can look at that both as these kind of heat maps as well as these kind of aggregate plots where this is essentially like aggregating the peak signal across all of the domains in the genome. You can see these sort of focal loop-like signals reform. And so again, we can do this with, this is the control condition, and then we do this in the presence of DTAG where we prevent or we degrade NIPBL, so we essentially prevent reloading of cohesin, and you see that the formation of most of the kind of structural features of TADs and loops are essentially largely attenuated, and we can see that both with these kind of genome browser shots as well as with these kind of aggregate peak type analysis effects where these kind of loop type signals really fail to reform. And so this kind of system really allows us to then go and ask, okay, during this kind of transition, what types of genes are actually requiring or require cohesin for their re-expression. And so to do this, we do these, this is using SLAM-seq. So we add these modified nucleotides that you can actually detect the nascent RNAs just through sequencing. It's sort of like a bisulfite sequencing type approach. So we add these for the 2 hours, first 2 hours during mitotic exit, and then quantify which genes are essentially upregulated or downregulated. We identify about 500 genes that are decreased in expression, about 85 that are increased. This is more than I think what's been described in the literature during sort of just acute cohesin depletion. But it's still not a huge number, right? It's still a relatively limited number of genes that are affected by this. And I think that we're actually fairly well powered because we did a similar experiment where we treat with things that inhibit RNA polymerase, and during the same time scale, we see over 10,000 genes fail to be induced. So I think that this kind of SLAM-seq type approach is actually really sensitive for detecting differences in gene expression during this time course. But I think the other thing that this allows us to do is to ask, okay, what are these genes that are actually sensitive to this? And can we identify any kind of patterns or features that might explain their responsiveness to cohesin here? So one kind of interesting thing that we can see is that if we just look at expression, so this is done in a slightly different way where instead of adding the modified nucleotides for SLAM-seq during the entire time course, we add them sort of in 15-minute intervals. And then we essentially quantify when is the sort of peak increase in expression of different genes. We actually see that the genes that essentially peak late during mitotic exit are the ones that are sort of most affected by cohesin removal. And I actually think that when you kind of examine the heat maps, this sort of makes sense that there's actually a fairly significant number of genes whose expression is really strongly reestablished before you have almost any semblance of sort of three-dimensional structure forming. And so I think one of the things that this suggests is that there's probably a large number of genes in the genome that don't really require sort of distal enhancer connections to be established during mitotic exit, but potentially just the decondensation of chromatin and opening up of promoters is sufficient to allow them to initiate expression. But for the subset that is NIPBL dependent, it is sort of expressed late, we can do things to try to examine what types of genes there are. And this is just sort of a simple Go terms analysis, but I think there's lots of kind of interesting terms that are suggestive of what classes of genes are coming up. So there's numerous terms that are related to sort of development, signaling, differentiation, proliferation, and growth. And so I think that one of the things that I think kind of arises from this is there's a decent number of genes that seem to be dependent upon cohesin in this context that are actually critical regulators of sort of like cellular identity and function. And so these are some examples that are involved in things like epithelial and mesenchymal transduction or signaling cascades and whatnot. So the other thing that we can see that I actually think is really cool and interesting is that when we look at like what these genes are doing in terms of their 3D organization, they're very clearly preferentially looping to regulatory sequences like enhancers, but in particular super enhancers. So these form actually sort of in normal cycling interface cells, these form sort of point-to-point loops with super enhancers, or they're clearly enriched for this. And we can actually see that they're also more likely to be associated with kind of super enhancer loci or super enhancer sites in the genome, at least in terms of genomic distance, which I think kind of fits this idea that these are sort of lineage determining genes that they're in proximity to the super enhancers, they may be more sort of super enhancer dependent. And so that I'm going to kind of summarize sort of the first part of the talk. So using these kind of degrons, I think we can show, which maybe we probably expect, that cohesin in NIPBL is really responsible for the formation of essentially most TADs and loops during the process of mitotic exit. That the genes that depend upon cohesin tend to be expressed later, essentially consistent with sort of a slight delay in the establishment of these kind of chromatin organizational features, and that these tend to be genes that are really related to lineage and identity. And these are associated with kind of super enhancers in a genome. And I think this is actually really interesting thinking about this idea of like mitotic bookmarking, because I think if you think about the idea of like inheriting some information from the prior cell cycle, one idea is you do this to sort of retain information about cellular identity. But I think from this standpoint, our data would argue that that's not really the way that mitotic bookmarking is working, at least at the promoter of these genes. These genes are really requiring these long-range connections to actually be fully established. And so we sort of think that if our data might suggest that if there's a role for mitotic bookmarking, it's really more in the sort of the rapid activation of genes during mitotic exit. Okay, so that was essentially the first part. I'm going to move on to the second, which is looking at CRISPR-Cas9 engineered structural variants. And this is the work of Jichao Zhu in the lab, who is sitting here in the front row, which is kind of a cool opportunity to present his work in front of everyone. Yeah, so a big part of kind of this aspect really was something we started thinking about essentially as soon as I started as a faculty fellow at Salk. And then this paper that I mentioned earlier from Stefan Munlos, I think had a big impact on our thinking, which is that, I think this is a seminal paper. It was really the first time that I think people could show that there were really clear phenotypic consequences to altering TADs and domains in the genome. And these impacts were really profound in terms of the sort of phenotypes. And so I thought this was really interesting, but also sort of thought, well, these types of events, right, deletions and inversions, these are rampant in cancer genomes, and this concept of sort of rearranging regulatory sequences in genes has actually been fairly well described in the cancer genetics literature, where a variety of different events like translocations, inversions, and whatnot fairly frequently will juxtapose oncogenes with novel regulatory elements, and this is sort of generally termed enhancer hijacking more recently. And so this kind of phenomena was sort of known to be a potential mechanism for oncogene activation. And we thought this was really interesting because at the time there was like huge volumes of whole genome sequencing from tumor patient samples coming online. And so we thought this could be a cool opportunity to learn more about where and when these kind of events were altering gene expression. And so one kind of wrench in this is, and part of this is we actually teamed up with Andy Futrial's lab, where they had done these whole genome sequencing studies they were part of this consortium called the Pan-Cancer Analysis of Whole Genomes Consortium, where they had whole genome sequencing from 2,500 or so patient samples. So they had thousands and thousands of structural variant calls, and we could essentially overlap these with Hi-C maps that we were making in the lab to say, okay, if you have a site where there's, say, a deletion that's deleting a TAD boundary, do you ever see genes nearby changing? And in this case, the reality is that, again, only a minority of these instances do you see any kind of change in expression. And so this is kind of similar to what we were saying in the first place. Why is it that some of these structural variants have these huge impacts on gene expression, huge impacts on phenotypes, but that the vast majority of these instances really don't have much of an impact at all? And so the way that we try to get at this question is basically using CRISPR-Cas9 genome engineering. And the way that we do this is we have like no clever, elegant strategy. It's brute force, basically. So we have two guide RNAs that we introduce into a cell along with Cas9, and we plate single cells. And we can see this experimentally that probably about 99% of the time, the two guides will essentially make cuts and be repaired locally. But in about 1% of cells, they appear to essentially repair across the breakpoint site to make a structural variant. And so that's fairly inefficient, but if you have good PCR primers, you can actually screen through these 96-well plates and you will find them. And because it's a completely new sequence that's being generated, it's actually fairly easy to identify them with PCR if you have good primers. And so we would do this, we would screen through stacks of 96-well plates. Jichao did a lot of this like during the pandemic lockdown, which was extremely challenging, I have to say. But so we can do this, we can grow up clones and do PCR, Sanger sequencing to confirm the breakpoint sites, and also do FISH to actually confirm that there are sort of large-scale rearrangements that have taken place. So the system that we have used for doing this is in these neuroblastoma cell lines. And the main reason for this is something that has been known in the literature for a long time. Neuroblastomas are sort of defined as this tumor where there's high levels of NMYC expression, and this is often frequently accompanied by NMYC amplifications. And this is the case for most of neuroblastoma samples, but there's actually a subset of neuroblastomas that are instead to have high c-MYC expression, and they tend to not have n-MYC expression. They tend to not have n-MYC amplification. So it's sort of like a mutually exclusive expression pattern. And in a paper that we had published a number of years ago now, we actually did high C in some of these c-MYC expressing neuroblastomas, and we could see they essentially all had these kind of enhancer hijacking sort of TAD fusion events at the c-MYC locus. And so we thought that this could be a cool system because of this sort of mutually exclusive expression pattern. And at the same time, the fact that these could, at least in this lineage, MYC could be activated by these kind of enhancer hijacking events. And so what we did is we took some of these cell lines that have high levels of the n-MYC paralog, but they have essentially no c-MYC at all. This literally has like 0 and I think 1 or 2 counts in the RNA-seq data, so it's about as low as it can possibly get. And they have no, structural variants, rearrangements, or anything like that at the c-MYC locus. So they're sort of like a blank slate. So we can introduce these kind of structural variants and really examine what impact they have. So what's actually capable of activating MYC or not. And so we do this where we take one guide RNA, we fix it near or sort of near the MYC gene. It's like actually 300 kilobases away, but it's in the same domain. And then we have a second guide RNA that we vary across the genome at different sites. We then do these experiments to screen through 96-well plates to find these events, and we can now subject them to a variety of different experiments. And so this is looking at high C data. This is from 2 of these engineered lines. So this is the one where the translocation goes to chromosome 7. This is one where it goes to chromosome 12. I guess I'll say, so looking at these plots, so this here is the high C data on chromosome 8, where MYC sits right here. This is the high C data on chromosome 7 that we've translocated this to. And then this diamond part in the middle is the high C data that crosses the breakpoint. So you see there's this kind of new domain that forms across the breakpoint. And so GChao actually developed a computational tool to examine this, breakpoint crossing high C data, say how often does this actually create a new domain? And it looks like basically in all circumstances, at least in these engineered clones, we seem to create some sort of new domain as a result of these translocations. So it seems like that's kind of the default consequence of this. But what's really interesting is if we look at expression, there's actually really widely variable patterns of expression of the MYC or c-MYC gene here. So this first part right here is the parental wild type cells. And so most of the clones, most of the events really do not activate MYC at all. But there's a subset that begin to activate it, and some of them activate it to a level that's actually comparable to some of the cell lines where we kind of find these events in the wild, I would say. And so what this allows us to do is really go in and begin to ask questions about, well, what distinguishes these events that activate versus don't activate MYC? And so early on, we were just kind of looking at some of these by eye, and we got an idea like, well, there's clearly something related to enhancer function in the partner region that's playing a role here. And so this is an example of this. So these are 2 rearrangements we made. This is one that goes to a region on chromosome 1. This is one that goes to a region on chromosome 12. And from the high C data, they actually look fairly similar. But the one that goes to chromosome 1 strongly activates MYC expression, and the one that goes to chromosome 12 really doesn't do much to expression. And so what we can see is if we look in the partner region, in this case at H3, K27 acetylation ChIP-seq data, I think this really begins to illustrate what some of the differences here. So this is a marker of regulatory elements, and in this case, like mostly active enhancers. So you have more enhancers, you have stronger enhancers, you have some of these that are classified as super enhancers. But the region on chromosome 12 has sort of fewer and weaker enhancers across the locus. And I would say that nearly all of the sites that we did these engineering have some kind of enhancer signal in the partner region. So it's not that this can't really just be explained by presence of enhancers versus absence of enhancers. It really seems a bit more related to almost the quantitative level that they're exposed to. And we can actually also go in and do experiments to really show that these depend upon the presence of the enhancers in the partner region. And so in some of these experiments, we had a fluorescent reporter in the 3' UTR of the MYC gene, so that when we make these rearrangements, these cells will turn green. And then we can use these to essentially go in and test like which enhancers might be driving the gene. And so this is an example where we delete this one enhancer. We see that in bulk cells, this causes a reduction in expression. And then we can actually go in and isolate like individual clones. And interestingly, we can actually see that the loss of expression is dependent upon the deletion occurring on the same translocated allele as MYC. So We actually can do this in a haplotype resolved way to show that this only really happens when the deletion is actually on the same allele. And so seeing this, we were interested in saying, can we actually develop any kind of predictive way of distinguishing events that actually activate versus don't activate MYC? And so we tried to essentially use predictive models based on this sort of initial idea of like how much enhancer signal is there to essentially distinguish these events that are activating MYC from the most of them that really don't. And so we tried kind of a few, you know, I would say sort of not particularly well-informed ways of quantifying sort of enhancer signal in the partner region. And the reality is none of these were actually very good predictors. So this is sort of these precision recall type curves, and you know, basically a random predictor is a diagonal line. So some of these were frankly performing worse than random. And so there seemed to be something missing. And so we went kind of back and looked at some of the events that we had made. And so most of the cases that I told you so far are instances where MYC is translocated to a new region and there's a set of enhancers that are nearby, which is sort of like this case I just showed a second ago. But in some instances, we actually made rearrangements where it was translocated to a new region, but there was a boundary that was actually separating MYC from sort of the bulk of the downstream enhancer signal that was sort of in the new partner site. And so this is sort of the example of one of these. There's a very small domain that forms here at the translocation breakpoint site. And then there's this large amount of enhancer signal that's then downstream from the boundary. And this enhancer that's almost immediately downstream from the boundary is actually the 5th strongest super enhancer in the genome of these cells. So we sort of think if anything is going to activate MYC expression, it's going to be this guy. But when we generate these clones and we do the RNA sequencing, we see that basically this doesn't touch MYC expression basically at all. And so I think this is kind of cool. It says, you know, the TAD boundaries here really do matter in terms of creating insulation in the genome. But it also, I think, more generally says that it's not just about sort of what enhancer signal is in proximity to a gene, but it's really how the gene is actually contacting these enhancers. And so this sort of evoked these kind of activity by contact models that have been introduced by Jesse Ingritz's group. And so in the kind of version that we're using here, we're looking essentially at a modified version of this where we're saying the expression of our gene of interest, in this case MYC, is going to be related to all of the enhancer activity that it's sort of exposed to, but where that's weighted by how frequently the gene is contacting each of these enhancers. And so the idea here is, you know, we have MYC, we have all these enhancers, and so you're sort of summing all of this enhancer signal across the partner region, but weighting it by what kind of high C contacts we see between the MYC gene and these distal enhancers. And when we use these now as a predictor, we actually see that this is much better at really classifying these events that activate MYC versus fail to activate MYC. And we can actually kind of go even further than this. We actually did this in response to a reviewer comment where there's actually been tremendous progress in recent years on using essentially deep learning to predict a variety of different genomic features. And there's very cool ways of doing this for predicting 3D genome organization. And so we used a tool by a guy named John Zhu, who Jijiao actually knows from high school, amazingly enough. He's like the star of the talk today. The idea here is that you essentially can train a deep learning model based on high C contact frequencies based on DNA sequence alone. And I think fundamentally what is going on here is that this is able to learn these kind of CTCF motif orientation rules. So the fact that loops will form between CTCF sites that have these kind of proper orientations with each other. And so we do this, we can actually compare these kind of in silico predicted maps with the ones that we actually generate. This is basically the predicted map. This is the site where the rearrangement is made. And this is, of course, in silico on the left, and this is the experimentally derived one on the right. You know, the in silico is trained on very high resolution data, so it's higher resolution than our actual experimentally derived one. And there are clear differences here, but I think it actually gets a lot of the major patterns right. It's finding boundaries in a lot of the places it should find boundaries. It's finding these kind of stripe-like features in places that these kind of stripe-like features emerge. And so, you know, I think that there's room for improving these, but in this case, I think this is actually doing a pretty good approximation. And if we actually include this now in these this ABC prediction, so instead of using the experimentally derived version, these actually perform almost as well or close to as well as using the actual experimentally derived high C data as well, which I think is actually really cool, thinking about like potentially using these deep learning tools to predict like the impact of structural variants in like an actual patient sample to see if you could learn in what instances these kind of events might be driving expression of different genes, like if you did whole genome sequencing on a patient tumor. So the other thing that we see, and this kind of relates back to this first part of the talk that I think is actually really interesting, is this kind of weird thing where there's actually not that many genes that seem to be all that sensitive to these induced rearrangements. And the way that we kind of got started on this aspect is we just looked at the other genes that were near MYC in the genome. And so MYC is in a relatively gene poor region of the genome, but there are other genes and in some cases annotated transcripts. And if we just look at the expression in the parental cell line versus these engineered lines, MYC is the only one that really changes appreciably at all. And it does so by orders of magnitude. And so we thought, well, there's some kind of crazy thing here where MYC is really sensitive to these changes in its local enhancer landscape. And we kind of wanted to understand more about this. And so the idea was really, well, maybe MYC is not alone. It's likely not the only gene in the genome that changes in response to its enhancer landscape in this context. And so we turned back to these kind of activity by contact models. So we said, well, we know that we can predict MYC expression. So let's see if there's other genes in the genome that we can find where the expression can be predicted by these kind of activity by contact scores. So what Jichao did is he took Hi-C data, RNA-seq data, and H3K27 acetylation chip-seq data from a panel of like about 20 or 30 cell lines. And you can calculate these activity by contact scores for every gene in the genome by essentially finding the enhancers that are nearby and calculating a score, summing that enhancer signal by how much the gene interacts with those surrounding regions, and then correlating that with gene expression. So we can do this across the genome. We can identify basically by randomly permuting the scores, identify sort of false discovery rates. And from this, we can say that we find about 900 genes in the genome that have these kind of significant correlations between these activity by contact scores and gene expression. And again, we can do these things like gene ontology to see, well, what kind of features and classes are popping up. And again, you see that there's a fairly clear enrichment of developmental genes, genes involved in signaling, and again, I think related to things like cell identity and function. And we can even go in and look at particular classes. So consistent with like developmental genes, we see there's a clear enrichment of transcription factor genes in this set, and as well as oncogenes, but I don't actually have this on the slide. These are all cancer cell lines. So I think that might explain part of the association with oncogenes here as well. And so using this, we can then go back to this original question of, are these more sensitive to these kind of changes in enhancer landscape? And does this explain some of these effects we're seeing of structural variants on gene expression? So we went back to this original data set from PCOG, where we have, again, whole genome sequencing from 2,500 patient samples. And we repeat this analysis of saying, if you're a gene that's in a domain in a patient sample that has a structural variant, how often do you see changes in expression? And now when we stratify these according to these sort of ABC correlated subset versus the rest of the genes in the genome, we see that this ABC correlated subset is actually significantly more likely to show changes in expression when it's found in the same domain as a structural variant, suggesting that these genes are more sensitive to these kind of changes in their enhancer landscape than most in the genome. And so we're very interested in this question of like why this happens and what might actually distinguish these kind of genes that are change in response to their enhancer landscape from the rest of the genes in the genome. And I think this is basically sort of my last couple slides here. The short answer is we can see some features that might be predictive of this, but we don't really have like a clear smoking gun of like a molecular feature that defines this. So we can see some features like they tend to be in considerably less gene dense regions of the genome. And that's quite clear. We can also see that they do this thing where they tend to essentially switch their compartment when they are expressed across these different cell lines. So this is from Hi-C data. And we think this sort of suggests they might actually move their position in the nucleus to some extent associated with gene activation or repression. But again, this is sort of like a feature, not really a molecular signature or anything like this. And we have looked for some things like they have a slightly higher GC content at their promoters, but there's not a huge difference here. And they are slightly more likely to have a CTCF site nearby, but I'm not going to like hang my hat on the differences between these two BioLen plots. So I think that we're still in the process of trying to understand really what are the kind of molecular features that actually fully distinguish these kind of classes of genes. But I think that's something that we're really interested in my lab in the future. Okay, so I'll summarize the second part. I didn't really have time to get too much into this, but we do see that there's clear evidence that structural variants have a fairly heterogeneous impact on gene expression. And that we think part of what explains this is that really not all of these structural variants are created equal. So there's clearly a relationship with this kind of enhancer load as well as the conformation of the partner region. But I think what this allows us to do is really try to begin to think about building predictive models to really assess or predict when the structural variant will have an impact on a nearby gene or whatnot, which I think has potentially really interesting applications for like personalized medicine type things in cancer. But I think, again, a very interesting, particularly from a kind of a basic science perspective, it's not really clear why, but not all genes actually seem to be sensitive to these kind of rearrangements of their sort of enhancer landscape. And I think this is really interesting, and it suggests that there are sort of different classes of genes in terms of their sensitivity to regulatory mechanisms in the genome, but we don't really have a clear understanding of what the molecular basis for this is. Okay, with that, I will stop and acknowledge the people who did the work. So the first part that was sort of the mitotic release experiments are essentially all done by Tessa Pope, who's a postdoc in the lab. And the second part was really led by Ji Chao Zhu, as well as Dongsun Lee, who was a former postdoc in the lab who now has his own group in Korea. We've had sort of tremendous support from a variety of different funding organizations, as well as really excellent support from collaborators, either at Salk or elsewhere. The imaging stuff was with Karen here from across the street. We actually had awesome help from Patrick Su when he was at Salk, basically figuring out how to screen through stacks of 96 well plates when we were doing CRISPR screens and whatnot. Yeah, and with that, I will stop and take any questions.
Questions? Let's start with you.
Not all these are set with the cohesion disruption, and there's hundreds of them actually have all the expression. And the second part, you could have not all these affected as so determined as ABC correlated. So I was wondering, do they have to be the same set of genes or what is overlap?
There's definitely overlap. I guess we haven't really just directly done the comparison. We did look, it's a little bit of a disappointment. We looked at one point to see, hey, could you make an ABC score that predicts the cohesion effect? And it doesn't really do a very good job. So I think that there's likely some component of ABC matters, but you probably have like a coefficient for each gene about like really how sensitive it is. Because if we just calculate like difference in ABC score, there's plenty of genes that have a huge difference in the ABC score that don't change at all. And so I think that there's additional information we would need to include other than just like a simple, how much does the contact with distal enhancers change? It doesn't work as well as I hoped it would for the first part.
Jesse, it's really cool to see a correlation between the so-called late mitotic actively activated genes and the dependency on cohesion. The question is related, well, twofold. One is, are these genes tend to be the ABC sensitive genes that you found in the second part of your or earlier part of your study? Is that right?
Yeah. So we actually haven't systematically compared. I think that there's clearly, just off the top of my head, there's clearly some that are shared. I think that there's also a component where what we see in the cancer cell lines is dependent upon lineage to some extent. So I honestly think that to make a really comprehensive thing like that, we need many more cell types, which, it's a great reason to generate tons of data.
A follow-up question, which is, let's say if you delay the depletion of cohesion after these genes are turned on, do you continue to see a dependency or not?
So that is an excellent question. And we have tried and are thinking about ways of doing this, but there's a big sort of caveat, which is that, I didn't really get into this, but the NIPBL cells, the depleted cells, they go into this very strong G1 arrest. So if we just degrade NIPBL, one of the things that we see, even at relatively short time points, is we begin to see cell cycle genes that are differentially expressed. But I'm not convinced that that's a direct consequence of like NIPBL on gene expression, but it has something to do with this G1 arrest that we induce. And I don't really know what it is. So I think that there's kind of a weird aspect where we're essentially trying to think about like, can we do this, but will we synchronize the cells in G1 either with like, you know, serum starvation or something like this, and then ask the same sort of question. But like we've tried to do some version where you basically just delay and then do this like several hours later, but we see tons of cell cycle genes that are coming up, which are not really the things that are in this initial set. And so it's not really an apples to apples comparison at that point. But like we've been trying to do some version of this now for like 6 months. Yeah.
Yeah. Yeah.
I mean, my hypothesis is it's something with the promoter region. But we don't have a ton of data that supports that. We did like motif analysis. We did these like GC content, you know, CPG island. We did like lots of sequence features of the promoter. Like when Jichao first showed me the ABC thing, I was like, it's CTCF. It's going to be at the promoter of these genes. End of story. Like write it up. And it was not. And so I think that there's some component of the promoters, either that they have some sequence features that we haven't really identified, whether they have sort of a lack of intrinsic activity, I think it's also possible, or that there's some component about them where they have a high off rate. So like some element of the promoter rapidly inactivates in the absence of a continued activation signal. But like, I don't really have a clear answer for like, what's the molecular determinant of that. Sorry, I can't hear. Yeah. Yeah. I think that that is an awesome idea. We actually wrote that in a grant that did not get scored very well. I have to say, I think it's a great idea. Yeah.
And then are involved in the gene function events that change how early or late that they activate?
Oh, that is a good question. We haven't, so the cells that we did in the first part are these RPE1, and so they're largely karyotypically normal. They don't have fusion events or whatnot. We could technically, well, we'd have to re-engineer the cells to look for the effects of like gene fusions or whatnot. I mean, I guess you could do synchronization and just look like a kinetic course. There's some studies out there, but we haven't really looked at that. But that is an interesting idea. Like if that, how, to what extent does, you know, changing the environment of the gene change its kinetics during the exit? But yeah, the stuff that we have right now isn't particularly well suited to address that.
Right.
Great, Jesse. I'm wondering, like a lot of things, and like cancer biology is talking about like interstitial heterogeneity, like these like developmental hierarchies where you get like stem-like cells or differentiated cells. Would you expect this model within like a tumor population where you have a translocation that enhances the moving on in one of these states that either more stem-like or differentiated state, so there's like kind of like a state-like effect on transcription within the for a given translocation?
I think, I mean, I think that there's probably an effect of intratumor heterogeneity. I mean, we can even see this in some of the engineered cell lines. Like the expression isn't like 100% uniform. Like you get subtle variation, and it's not clear to what extent that might be like karyotypic instability, like it's losing the allele over time, or it's actually there's some effect where if it initially rapidly and strongly establishes contact, it maintains those over time. So I think it's totally possible. The flip side that I think would actually be really interesting that we haven't really looked into, but I think would be cool is, because the cohesin is so dynamic on chromatin, I could easily imagine a world where that actually also contributes to some of these intratumoral heterogeneity effects of let's say you had a loop forming at EGFR in, half your sample, but it fails to form the other half. Well, that might actually contribute to sort of intratumoral fluctuations in expression and whatnot. So there are these studies where, like Arjun Raj has this stuff about 5% of cells will express some oncogene, but it's not really clear what the basis of that is in culture. So I think looping could have an effect on that, but we've never really done things to look at that. But
Maybe one more question.
Yep.
Sort of going back to promoter features that we flag in for current association or something, however you want to describe it. I think that interesting approach, there's recently a pretty impressive model of basically promoter page data, so sort of sequence.
oh yeah, yeah.
That are there as making driving most of the I wonder if you can look into a model like that, maybe not even necessarily or create your own data set.
Yeah. I think that is a very interesting idea. If I remember right, in that paper, they have something about, it's some of the motif classes are, well, there's some component where some of the motif classes seem to respond more to context, like genomic context, which I could imagine would be something like whether there are enhancers nearby or whatnot. I don't remember the details of it, but no, I think that would be actually, we have not done that, but that would be a kind of a cool thing to look at, is the features that they identify and how that plays in. Yeah.
Great. Okay, thanks so much.
Thank you.